Tom spent 15 years doing what Kate and I have done, worked as scientists in diagnostic labs, and completed a PhD part-time at UWA. Tom was appointed Senior Medical Scientist in charge of the Department of Clinical Microbiology at Charles Gardner Hospital in 1988. In 93 to 94, he undertook a master's degree in applied epidemiology at the ANU. In 1995, was appointed an associate professor at the Department of Microbiology at UWA. He was also principal research scientist at the Australian Centre for Pathology and Medical Research, now Pathwest Laboratory Medicine. In 2002, he was awarded a personal chair at UWA. Has a long-standing interest in healthcare-related infections, particularly the diagnosis, pathogenicity, and epidemiology of Clostridium difficile, and the antimicrobial activity of Australian native animals. So if you like tea tree oil, go and see Tom. He loves it. He's a fellow of the Royal College of Pathologists, the Australian Society for Microbiology, and the American Academy of Microbiology, and the Faculty of Science of the Royal College of Pathologists of Australasia, and published over 250 book chapters and refereed journal articles. Professor Tom Riley.
Well, you could give me a long one.
Oh, you didn't have to read it all out. Now, if I stand over here, I kind of like to walk around, but I'm a bit worried that the cam. Oh, will it follow me? Oh, excellent. Can we go back to that slide that was? Hello, Mr. Slide Man. Can we go back to the one that was on before? Your little, no, not that one. The next. So I thought this was fantastic. Something old. We've just been talking about TB, which has been around probably since Adam was a boy. And you know, they've found TB in mummified Egyptian kings. So it has been around for a long time. And so now we're going to talk about something relatively new, certainly in the microbiology field. So if we can go to the next one, and then I can, or can I do that myself? Oh yes. Was that me or you? Fantastic. Clostridium difficile clearly has been around for a lot longer than the last 30 years, but it's really only come to the fore in that 30-year period. I was excited to see all this alcoholic hand gel on all these tables, and if there's at least one take-home message you get from this talk, this is it now. It's no good for Clostridium difficile. So we can. I don't actually have a lot of infection control stuff in here because dealing with C. difficile is actually quite simple. Very complicated disease. Pathogenesis of infection is very complicated, and I'll talk about that. And I'll try not to be too complicated, but I am going to talk about whole genome sequencing and some other fairly high-powered things. So pathogenesis, diagnosis is even complicated these days, but dealing with it's actually very, very simple. If you follow, and Madeline will tell you this, follow simple infection control practices, and you can deal with Clostridium difficile. So this is the sort of problem. This is the most common cause of infectious diarrhea in hospitalised patients. The bigger problem that we're facing now, which is what we'll eventually get round to in this talk, is that this is now an enormous problem in the community. There is a lot of C. difficile diarrhea in the community, and you will see patients that have C. difficile diarrhea. Now, they might not know it, their GPs probably won't know it, and that's one of the issues that we're trying to deal with at the moment. And this has all happened really in the last couple of years. So in that regard, this is very new. So being a Clostridium, Clostridia cause disease because they produce toxins. So tetanus produces tetanus toxin. Clostridium perfringens produces nasty toxins that cause gas gangrene. Botulinum produces botulinum toxin. So all the Clostridia produce these really potent exotoxins, and that's how they cause disease. And Clostridium difficile is no exception. It produces these two major toxins, we're not very smart in microbiology. Toxin A, toxin B. We haven't found toxin C yet, but this probably will get called toxin C. So this is the third toxin, which was first described by a funny little French man named Michel Popov in, that's his real name too, in and you cannot understand him. He's about this high. He speaks with a very thick French accent when he's speaking English, and I can never understand a word he says. But he described this in a French isolate in 1988, and then we all forgot about it. But you'll see when we get towards the middle of this talk how relevant binary toxin is. So C. difficile sprang to the fore in the mid-1970s, when patients in the United States where clindamycin had suddenly come onto the market. You've got to remember that in the United States, metronidazole was never used initially for treating anaerobic infections because they thought metronidazole caused cancer. So no metronidazole used in the United States. The only thing that was used for anaerobic infections was clindamycin. Good drug, good for staph and good for a whole bunch of other things, but was always used for anaerobic infections. And so as soon as this came onto the market, so clindamycin is the 7-chloro-derivative of lincomycin, marketed by Upjohn, all of a sudden we have all these patients developing diarrhea post-clindamycin therapy. And indeed it was called clindamycin-associated diarrhea. A proportion of those patients then went on and got pseudomembranous colitis, and patients with pseudomembranous colitis, which I kind of call diphtheria of the gut, where you get this membrane forming over the gut. I may have a photo, I can't remember if I put one in. But the only thing you do with those patients is you cut out the affected portion of the gut. And given this is a disease that primarily affects old people, having major surgery is not without its risks. So we had this issue of clindamycin-associated colitis. It appeared to be transmissible because of outbreaks of disease, and there was a lot of effort went into trying to work out what the problem was, and eventually, even though some people thought it was a virus, it was shown that this was actually caused by Clostridium difficile. So these two toxins, and toxin B, strangely enough, was the first toxin to be described, and I'll show you the effect of toxin B on tissue culture cells in a minute. Toxin B is a very light, well, they're both very heavy toxins, but toxin B is lighter than toxin A. So if you put both toxins through a column chromatograph, the very heavy toxin, which is toxin A, comes out first. So that's why, even though toxin B was the first toxin to be discovered, toxin A is called toxin A and toxin B is called toxin B. So these are these interesting toxins that are the genes that encode these toxins live on this part of the chromosome which we call the pathogenicity locus. And this is a diagram of the pathogenicity locus. Here's the gene for toxin B and the gene for toxin A. And there's a couple of regulatory genes. So this gene here upregulates toxin production, so it causes the bug to make toxin. And this gene here downregulates toxin production, so it stops toxin production. Now, I will be asking questions about this later on, so you have to remember this bit. This one's the most important, downregulation, because what I'm going to talk about in a minute is some mutations in this gene so that it doesn't downregulate. And so the bug keeps producing more and more toxin. That's really all you need to know about this, other than that both these toxins affect the actin cytoskeleton. So the thing that gives you shape is actin inside the cell, and it allows the cell to be nice and round. And so what these toxins do is cause that actin to depolymerize and the cells just collapse. And so when that happens to the cells that line your gastrointestinal tract, you get loosening of the tight cell junctions. And so the diarrhea that this organism causes is not like cholera or enterotoxigenic E. coli, where there's a toxin that mainly activates adenyl or guanyl cyclase. So it's an active process of pumping out water into the lumen. All this does is cause those tight junctions to part and fluid just pours into the lumen. So even though we call this an enterotoxin, it's not really an enterotoxin. So And then we have this other toxin that my funny little friend Michelle described, and this toxin is also a toxin that affects the actin cytoskeleton. It's got a very fancy name. It's an actin-specific ADP ribosyltransferase. I won't be asking any questions about that. But 30 years ago, we would say that maybe 2 to 5% of isolates of C. difficile we could find produce this toxin. Currently in Australia, it's about 12%. So, and it's interesting that strains of C. difficile that produce this binary toxin seem to produce more severe disease, and that's important. The other thing that's important to know is that strains of C. difficile that come out of animals, other than humans, invariably also produce this binary toxin. So I'm going to talk a little bit about animals as we progress, because animals, production animals, and maybe companion animals, are central to this story. So you can see sort of cytopathic effects, and we won't worry about the writing, we'll just look at the pictures. So this is a cell that's nice and flat and lying flat on the glass that it's attached to, and when you put some toxin on it, it rounds up like this. It doesn't kill it straight away, but eventually it will die. And then when you look at it histopathologically, when you take out somebody's gastrointestinal tract and cut sections, you get these things here, which surprisingly are called volcano lesions. And you can see why they're called volcano lesions, because that's exactly what they look like. And so this could be a human, but equally, if I did a necropsy, because we don't do autopsies on piglets, we do necropsies on piglets. If I did a necropsy on a piglet that had been infected with C. difficile, you'd see exactly the same thing in a piglet. You see these volcano lesions. And that's because you get this massive inflammatory response, you get lots of polymorphs, and then you get that increase in epithelial permeability that I was talking about before. So all these things here, you can't see it very well on this picture, but there's lots of polys in there, and all these inflammatory cells have been attracted to the site. So eventually the whole gastrointestinal tract gets covered, or the large bowel in the human case anyway, gets covered by this membrane. And clearly in that situation it doesn't work very well. So now we're going to talk a little bit about genetics. So this is a phylogenetic tree, and what it tells us is that if you take all the C. difficiles in the world and look at their genetic makeup, you can divide them into 5 different groups. And again, we're not very smart microbiologists. The 5 different groups are labelled 1 to 5. So this is group 1 down here, which is the most common. This is group 2. This is group 3. There's only a couple of representatives in this particular tree. And this is group 4 out here. And then way out here is group 5. And we actually call them clades, but clade's just a fancy scientific word for group. So this group here, this group 5, this clade 5 up here, is a very interesting group, because in evolutionary terms, the distance from there to there is about 80 million years. And can anybody tell me what happened 80 million years ago? I know many of you weren't there, but nobody knows. Who? No, it's when Australia separated from the rest of the world. It happened 80 million years ago. And so when you start looking at strains of Clostridium difficile from Australia, they all fit in to this group. And they're completely different to strains of C. difficile we see in other parts of the world. Now this is kind of early days in this sort of investigation, but this clade here, for example, which is clade 2, is a North American, might be South American too, but we don't have any data from South America. So at least it's an American clade, and possibly a North American versus South American. This particular clade here, which is clade 4, is an Asian clade. So there's certain strains of C. difficile within those clades that we can identify as having come from that part of the world. And so if they turn up in a different part of the world, something's got to be happening. How did they get from A to B? So just bear that in mind. This will all come together nicely, we hope, towards the end of the story. So now let's get that back to a few more basic issues. The reason why your alcohol hand rub doesn't work particularly well is because Clostridium difficile forms spores. And those spores are very resistant to heat, drying, and alcohol. And in fact, in the laboratory, we use alcohol to select for C. difficile spores. So don't use that if you've got patients with C. difficile. Wash your hands properly. Not that soap or any of those other things are going to kill your spores, but it's just the physical action of washing your hands, if you do it properly. Madeline can tell you how to do it properly. That will get rid of C. difficile spores. And I can tell you that C. difficile gets everywhere. It contaminates everything. And so it contaminates the hospital environment. But not only that, it contaminates the external environment in a way that you'll see in a minute. So if you're going to get C. difficile, first of all, you've got to get exposed to the organism. And the infectious particle is the spore, but we don't actually know, 30 years after this was first discovered as a cause of disease, we don't know how much C. difficile actually is an infectious dose. For many organisms, Shigella dysentery, for example, somewhere between 10 and 100 organisms is an infectious dose, and that will cause an infection. With Vibrio cholera, it's in the thousands. Vibrio cholera is not a particularly pathogenic organism. For C. difficile, we don't know, and that's a bit of a problem now because I'm trying to work out how are these people in the community getting infected, via what mechanism, and how much C. difficile is required for them to ingest. The complicating issue, of course, is the other risk factor, and the other risk factor is, I guess I've put exposed to antibiotics, but really it should be exposed to anything that changes your gastrointestinal flora, because you won't get C. difficile disease unless you've got an abnormal gut flora. So your gut flora is protective. You don't get C. difficile disease if you've got a normal gut flora. So antibiotics are the things that clearly change your gut flora most, and they're the most common things that change your gut flora. And there are other things, of course. You know, some cytotoxic drugs will do it. If you have a norovirus, rotavirus, viral diarrhea, that will change your gut flora. If you have a salmonella enteritis, that will change your gut flora. So anything that changes your gut flora, but the most common thing is exposure to antibiotics. So the difference with this organism in terms of pathogenesis, it's kind of this two-step process. You've got to have something that perturbs your gut flora first, and then you've got to get the infective dose. So it's much more complicated than what some people give it credit for. Now, of course, if you're very young, and you're a very young human, or you're a very young pig, or a very young horse, or a very young cow, you don't have a gut flora. So very young individuals, neonates, are exquisitely susceptible to colonisation with Clostridium difficile. And all, as far as I can determine, warm-blooded animals, I shouldn't say all because we haven't looked at marsupials yet. We're about to do a project on marsupials. But certainly all mammals get colonised by Clostridium difficile in the neonatal period. You go and look at them a month later, no C. difficile, it's gone, because all the other gut flora comes in and it kicks out C. difficile. So Clostridium difficile is a bit of a wimp. It doesn't like competing with other bugs. And clearly that's, you know, part of its overall strategy for survival, its natural history. All it wants to do is colonise somebody's gut for a few weeks, multiply up, get squirted out all over the ground. We change that with the human thing, but you know, for most animals, squirt it out on the ground and then infect the next animal that comes along, or the next baby. So when you look at the data on colonisation of human neonates, you see the colonisation rates differ dramatically. Some papers say 0% of kids get colonised, some papers say 80% of kids get colonised. And it really amazes me that people can't understand why there's this difference. it's all a reflection of the environment into which these kids are born. If you're born in a brand new hospital, when King Edward's eventually rebuilt, right where my old laboratory currently stands, brand new hospital, no C. difficile in the environment, none of those kids are going to get colonised by C. difficile. You're born in a dirty hospital, then you're going to get colonised by C. difficile. But as I said, if you don't interfere with nature, then it will get kicked out in a month's time and never be seen again. The problem is, of course, we do interfere, and the way we interfere the most is by filling up these animals, and indeed the humans, with antibiotics. So that's kind of how C. difficile gets into the body and what happens. And you can see the effect of antibiotics on this slide. It's not so that you take a course of antibiotics, your gut flora is messed up, you stop taking your antibiotics, the next day you're fine. That's not true. It appears that at least 2 months after you've stopped taking antibiotics, your gut flora is still perturbed, possibly 3. Some people are now saying 6 months. So during that period, you're susceptible to infection with C. difficile. Now, the risk is greatest in the first month, but there is still a risk 6 months out before your gut flora normalises. So anything you can do to help that process, and this is where the probiotic thing comes in. I believe that anybody taking antibiotics these days should take a good probiotic. The trouble is, it's hard to tell what's a good probiotic because half of them are dead and don't do anything. But if you can find a good live probiotic for patients on antibiotics, that's a good thing, and they should keep taking those probiotics for several months after they've stopped their antibiotics. I can't give any plugs to any probiotic manufacturers or whatever, because but there are some dodgy ones out there. That's the problem. So I mean, something I will give a plug then. So Yakult, it's, you know, Japanese, and it's Lactobacillus casei, whatever it is, Shirota strain. I think Yakult's pretty good. There's a Lactobacillus GG, which is also a Lactobacillus casei, but a different strain. It's the Rhamnosus variety, and the 2 Gs, you'll see that in Valia yogurt, I think. So the 2 Gs in Lactobacillus GG stand for Sherry Gorbach and Barry Golden, and it's one of them produced the specimen from which the Lactobacillus came, and that's been commercialised worldwide. So as long as you find a viable probiotic, it's good. So that's very important. You are no longer not susceptible to C. diff when you stop taking that antibiotic. It takes a while. So I've been working on C. difficile now for 35 years, I think. Madeline was a small child when I started, and so and literally for 30 of those 35 years, nobody's given a rat's ass about what I've said, until this sort of thing started happening in the United States about 8 or 9 years ago, when all of a sudden we started to see these huge outbreaks of disease in North America and in northern United States and southern Canada. And we all thought this was one huge outbreak of the one strain, because this here says it's predominantly clonal. So this is the, these are all Canadians, I think, from memory. Yes, these are all the Canadians talking about their outbreak in Quebec province, and the strain of C. difficile caused this outbreak was called the Quebec strain. Same thing was going on in the US, centered around Pittsburgh, Pennsylvania. And for years we thought it was actually the same strain, but in fact there was 2 completely separate strains arose. That Quebec strain moved to the United Kingdom, and there are a number of quite well-publicised outbreaks, the first of which occurred at the Stoke Mandeville Hospital. And if you're interested in reading this report, this is a fantastic report on this big outbreak and it describes all the things they did wrong, where hospital management kind of rode roughshod over infection control people and said, oh no, we've got to move these patients all over the hospital. It doesn't matter that they've got diarrhea. And so in this particular outbreak, there were two different times of over about a two-year period, approximately 300 cases in each outbreak. I think from memory, 50 or 60 deaths in each outbreak. And so this report is a really good report to read. The hospital management or trust manager got sacked over this, and there were several other big outbreaks in the UK at around about the same time. All because basic infection control was ignored. So started in North America, spread to England and then across the Channel. Became a real political issue in the United Kingdom because of these sorts of things, where people were dying. I think somewhere down here. Oh well, yes, there's 2,247 deaths, whatever year that was, 2007, 2008. So let me go back to Canada for a minute and try and put this into perspective. So in Quebec province is about the same size as New South Wales, about 7 million people. In 2004 in Quebec province, they had 7,004 cases of C. difficile diarrhea, and they had 1,270 deaths. So that's 20 people a week dying in Sydney. And people wonder why I jump up and down about this, because we are at risk of something exactly the same as what went on in Quebec happening here in Australia. Because the history of the outbreak in North America, even though we don't really know the history of the outbreak in North America, but that's exactly what's going on here in Australia now. We're about 10 years behind what happened in North America. This became a very political issue in the United Kingdom, because once the tabloids get onto it, all the politicians get worried, questions get asked in Parliament, and a very nice man by the name of Andrew Pearson was put in charge of, first of all, establishing surveillance for MRSA in the United Kingdom, and then on top of that, for C. difficile. And there's a paper came out, the MJA only last week, saying that the number of deaths associated with C. difficile in the UK now is really at record lows. And Andrew Pearson did not report to the health department in the UK. He reported to the Prime Minister's office. And so we're talking about Tony Blair era. So Tony Blair made this a political issue, and they spent money, and they spent lots of money, improving infection control in the United Kingdom, and eventually it had an impact on their rates. Unfortunately, out in the colonies, so here we are in the United States, things are very grim. So whereas in the United Kingdom and Canada have got their situation pretty well under control, in the United States, it's completely out of control still, because healthcare in the United States, as I'm sure you're aware, is completely dysfunctional. And so you get very, 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 very good hospitals, but you get a lot of very, very, very bad hospitals. And so this is a paper that came out a couple of years ago now. Luke Chen's actually Victorian, so he's Australian. We've got a bit of Australian content. But this is just saying that C. difficile in this group of community hospitals, very important to remember, community hospitals, C. difficile is, or has replaced MRSA as the most, hi Stephen, as the most common healthcare-related infection in the United States. So this is huge. This is $3 billion a year costing the American healthcare system at the moment. And if you go to some of these American hospitals, that particular outbreak strain, which I'm going to talk about in a little bit more detail in a second, it's still 60 to 70% of their strains of C. difficile that they're isolating in their hospitals. So it's causing huge problems. And this is the particular strain. So this is called PCR ribotype 027, and cause more severe disease. It has those, remember that funny regulatory gene that I showed you way back when? it has deletions in that gene, it produces more toxin, it produces this binary toxin. None of that probably caused it to spread, but it also has a mutation which caused it to be fluoroquinolone resistant. And in the United States, unlike here in Australia, fluoroquinolones, particularly a drug called levofloxacin, which we've never had here in Australia, fluoroquinolones were the recommended drug for community-acquired pneumonia. So you had all these people getting this drug called levofloxacin, and this strain of C. difficile was resistant to that drug. And so that's what caused it to spread across North America. Big epidemics. As I said, numbers dropping in Europe is still a major issue in the United States. Now we've had three, we can call them clusters. We've had at least four introductions into Australia. The first patient with an 027 in Australia we discovered here in Perth. It was a patient of Clay Golledge's. She had just returned from the United States. She was taken off the plane. She got sick in the US, decided she would try and travel home. She was a bit unusual. She had had a colectomy for ulcerative colitis, so she kind of wasn't normal to start with. She was taken off the plane in Hawaii, spent a week in intensive care in a hospital in Hawaii, was presented with a bill for, I think, $300,000 when she got out of this hospital, and her insurance company refused to pay because they said, oh no, it's pre-existing illness. But in fact, we had been monitoring this girl for some time, and we knew she didn't have C. difficile when she left the country, and she had C. difficile when she came back. And anyway, eventually they coughed up and paid the bill, so that was a good outcome of a bit of forensic microbiology, I guess, if you like. So, but we were lucky. We had no spread here. I mean, she was actually treated as an outpatient here. We had no spread that we know of, and that was a bit of a bullet that we dodged. We have had 3 other introductions into Australia, and you can actually see them. So this is a little bit more of that sequencing. So this is sequencing the whole genomes of these strains. And just to show you, these 2 little red dots up here, these are a bunch, or this is a bunch of strains from Melbourne. So this was the first actual introduction of this organism into Melbourne. And this just makes the point that there are 2. Remember I said this strain, this 027, was fluoroquinolone resistant. There are 2 different mechanisms of fluoroquinolone resistance. And so the strains that we found that came out of Pittsburgh formed one group, and then the strains that came out of Canada formed a second group. And so we know that our strains that end up in Melbourne are related to these Canadian strains. And so you can see that on this picture here. So this is just showing how these strains have travelled the world. So the American strains didn't travel very far, because you know what Americans are like these days. They don't like travelling because they think they're going to get blown up. But the Canadian strains have travelled in and out of Europe on many occasions, and then eventually, so this is a paper, if you're interested in reading Nature Genetics. It was published a couple of months ago in Nature Genetics. I did try and tell Trevor Lawley, who was the senior author, that this is not where Melbourne is, but unfortunately, Trevor's actually a very nice Canadian chap, but his geography is not real special. So we could trace the importation of our strains, initially our very first cluster of strains, to the Epworth Hospital. I can say that because I think it's public knowledge now. And all the people at the Epworth were a bit strange about this, because they said there was no evidence of infection control in the Epworth Hospital, and that there were 6 or 7 patients. And they said, no, it must have been 7 or 6 separate introductions of this very rare strain into their hospital. So we've actually gone on, we've done whole genome sequencing of all those strains, and the beauty of doing whole genome sequencing is that you can tell exactly whether a strain is the same as the strain in the patient next door. Much better than all this other typing that we do, this pulse field gel electrophoresis that most people do these days. If you do the whole genome, what you find is, so here's the Melbourne strains down here, 1, 2, 3, 4, 5, 6, and you can trace them back to Canada. And so that was the beginning of 2010. At the end of 2010, we had this big cluster of same ribotype 027 strains in North Sydney. And the people in Melbourne said, that's where it must have come from. It must have come from North Sydney. And the people in Sydney said, no, 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 you must have introduced it from Melbourne into North Sydney. So in fact, we've also sequenced a bunch of strains from Sydney. And you can see the Sydney strains actually came from the United Kingdom. They came from the Hammersmith Hospital and were introduced into northern Sydney. And in fact, this part of northern Sydney, there's a very big, I shouldn't say this because I know there's some people from the mother country here. There's a big British contingent live in this part of northern Sydney, and they've imported this ribotype 027 into northern Sydney. So Melbourne didn't give it to Sydney and Sydney didn't give it to Melbourne. They've both got this independently. So this is the beauty of doing this type of typing. So we had those two introductions. There was a bit of spread here. So this 027 in Melbourne went to a nursing home in Frankston because patients go from hospitals into nursing homes or aged residential care. Sylvia Gandossi will be very upset with me for calling them nursing homes because I believe we're not supposed to call them nursing homes anymore. So they're the only 3 clusters of cases we've had in Australia at the moment of this particular strain. So that's not what I'm concerned about. I'm more concerned about the introduction of new strains and us breeding our own nasty strains. So C. difficile infection in Australia, it's not notifiable, and so that makes it quite difficult for us to keep a handle on exactly what's going on, although we do have mandatory reporting by hospitals since 2010. And so what hospitals do is they report what's called hospital identified cases, which is supposedly then translates into the burden of disease that hospitals have to look after. And the problem with that, of course, is that it's not a good, doesn't give you a good handle on what's going on in the community. And that's, I think, where we have this problem at the moment. So if you have a look at what happened to C. difficile, as I said, I've been interested in this for a long time. So this is Charles Gardner Hospital data, and you can see in the early 80s, if you look at the blue line, numbers went up quite dramatically to the stage where our rates were about 7 cases per 10,000 occupied bed days. And that was all driven by cephalosporins. So Clostridium difficile is intrinsically resistant to cephalosporins. And if you give a patient cephalosporins, which have very broad spectrum, you will be preparing them for colonisation and infection with Clostridium difficile. And so then at Charlie's, for a completely different reason, in 1998, by that stage we were only using ceftriaxone, great drug, but it was being overused. It was a race when you got to casualty, whether you'd get your ceftriaxone or your CAT scan first, one or the two. And clearly this was inappropriate use of CAT scans as well as ceftriaxone. And so the Drugs and Therapeutics Committee was actually concerned about multi-resistant gram-negatives. They weren't worried about C. difficile, but they banned ceftriaxone. Ceftriaxone was pulled from the formulary. A little bit was left in casualty for patients with meningitis. And you can see, I think here, well, all here, these are the rate of C. difficile plummeted once we pulled ceftriaxone. And it went down to less than 2. In fact, it went down from 2000 to 2010. The rate at Charles Gardner was about 1 case per 10,000 occupied bed days. So very low rates. And you can see this is a time series analysis which shows the same thing, except we've got a control. So the blue line is Royal Perth. As you can see, here's the period, this is the time when we stopped the ceftriaxone. Blue line is Royal Perth, and the blue and yellow lines, the yellow being Charlie's, crisscross here. When we have the intervention, Charlie's rates go down, and Royal Perth Hospitals kept going until they had their VRE problem, which was also due to cephalosporin use. And I do have a slide which says cephalosporins are the work of the devil sent to end the antibiotic era, and they should be banned. So what's been happening since 2010, and this is where it gets to be quite worrisome. So these are that mandatory reporting data that goes to the Safety and Quality Commission. And you can see the blue line's the overall rate, and this is the tertiary hospital rate. And you can see here back in beginning of 2010, here we are, a bit over 2 in the tertiary system. We have this little blip here. And at about that time, we started changing our testing procedures to a more sensitive testing procedure. And then we have a good at least 6 months where things plateaued again. And then from the middle of 2011, the rates have gone up and up and up. Now, interestingly, the grey line here is the metro non-teaching hospitals. So that's things like Swan Districts, where what's her name comes from? Marilyn Hassel comes from. Swan Districts and Armidale, and I don't know if Joondalup gets a gig there or not. But very interesting, you see, 4th quarter of 2010, peak, 4th quarter of 2011, peak. Now, I don't think that's an artefact, I think that's real, and I think there's a very good reason for it. But we started seeing changes in the tertiary system about the middle of 2011, or certainly the 3rd quarter of 2011. And so if you want to see what happened in terms of numbers, I mean, these are the numbers, and that kind of really, I think, brings it home that we've seen lots more patients with C. difficile infection in Western Australia. And there's a hint, I call it a hint anyway, of seasonality. There's a little peak there, I think, and there's clearly a peak there. So that's up to the middle of 2012. So it went up, but then it came down. So why did we see this increase? Why did it drop? What's going on? And this is something which has occupied far too much of my time for the last 2 years. There are various reasons why you see increased numbers of cases of any sort of infectious disease. So one of them is maybe people are doing more testing because of greater awareness, and there is some evidence that that is true. I've been getting up like this for the last 2 years, banging my gums on about C. difficile and how important it is. Maybe it's having some impact. We have, as I mentioned, changed our testing methods to a more sensitive method of testing. But neither of those two things really account for the increase that we've seen. We've seen a 250% increase in the number of cases in the last couple of years. So if it's real, why is it occurring? And is it just healthcare associated? And what's the role of the community and other changes in risk factors? So community C. diff is not new, even though I'm going to show how much it's increased in the last couple of years. It's been around for forever. People get C. difficile in the community because patients get antibiotics in the community. Every year we have this huge seasonal peak in antibiotic use at the end of the winter cold flu season. Generally speaking, though, there's no source of C. difficile in the community like there is in the hospital. We amplify C. difficile in the hospital gets contaminated, and patients then come into contact with those C. difficile spores. So maybe there's been some change in the community so that patients are coming into contact with C. difficile. So the most obvious source are these animals that I've been talking about before, cats and dogs and horses and pigs and cattle. Now, we don't often come into contact with cattle or pigs unless you're a farmer, but you certainly come into contact with the meat products that come as a result of that. So this is what we've been looking at for the last couple of years, is trying to make a link between what's going on in the production animal industry and what we're seeing in humans. So if you go back to these tertiary, non-tertiary, I should say, non-tertiary metropolitan non-tertiary hospitals, so the Swan Districts and the Armidales, and look at their pattern of disease for this 2-year period, you can see quite clearly that there was a peak at the end of 2010, and there's a peak here. But interestingly, if you look at whether or not these patients had community-acquired disease versus hospital-acquired disease, in the community hospitals, 45% of the cases are actually community-acquired infection versus 25% community-acquired in the tertiary hospital system. And that kind of makes sense because if you've got a bit of diarrhoea, you're not going to go to a large tertiary hospital first. You're probably going to go to a smaller hospital. But of course, the obvious thing from that is that really what you're going to do first is go to your GP if you've got diarrhoea. And the problem is we're not capturing that group of patients at the moment. So you can tell that these are different. So if you get hospital-acquired C. diff infection, so this is the same group of hospitals, metropolitan non-teaching. So this is the age distribution for hospital-acquired disease. So that doesn't change whether it's metro non-teaching or tertiary hospitals. Looks just like that. But if you look at community-acquired disease, it's completely different. So you get lots more younger patients who get C. difficile disease in the community. So what's going on? And it's not just us. This is South Australian data. And again, you can see is peak. This is their 2011 peak, and this is their 2012 peak. This has been going on right around Australia in every state. We've seen this huge increase in the last couple of years. So what happened further down the track in Western Australia? So here's our little bump, end of 2010. Here's our big peak, end of 2011, beginning of 2012. Came down, went along, and it's gone up again. So we're seeing this now, seasonality of C. difficile infection. So why is that? That's the $64,000 question. Well, so what we showed was that much of that increase has been due to the arrival of another, not another strain. We've never had much of this O27 strain, but its kind of first cousin has arrived, if you like. So this is a strain which we call ribotype 244. Does exactly the same sort of things as that ribotype 027. And as I said, by genetic sequencing, they have come from a common ancestor. So this strain has suddenly appeared, it's causing community disease, it produces more toxins, produces that binary toxin. And when we did our very first Safety and Quality Commission survey back in 2010, we had one isolate in 350 isolates that we looked at. And then in that 2011-2012 period, this was the third most common ribotype circulating around Australia. So it's gone from nothing to being the third most common ribotype in the matter of 12 months. And so this causes, as I said, much more severe disease. So where's it coming from? I'll skip over that. We'll get to the bit about where it's coming from in a second. So if we do more of this genetic sequencing, so what we have here, well, you can probably read most of these. So here's a patient from Charles Gardner, here's a patient from Fremantle, but here's a patient from Southern Health in Victoria, and one from John Hunter, one from the Austin, one from Mona Vale, which is just north of the Royal North Shore in Sydney. And this is whole genome sequencing. So by whole genome sequencing, these strains are virtually identical. So how can it be that a patient in Perth and a patient in Sydney has exactly the same strain of C. difficile? There's only one logical explanation, and that is that they've got it from a common source. And my problem at the moment is trying to determine what is that common source. The most likely sources of infectious agents when you've got a common source outbreak is food. So when you look at the strain, it's this ribotype 244, and as I said, it's a close relative of that ribotype 027. So in fact, we will go back to this. So this is that same structure of groups that I showed you earlier on, but just different. So this is that group 1, clade 1, here in red. is clade 2, and that's a 244 there, and that's an O27 there. What did I say? I said that is a North American organism. So if it's food and the bug's North American, it's got to be North American food. So how many of you know what we import from North America in terms of food? You must go down to your local Coles and Woolies. Very good. Oranges. What else? No. Well, but in large quantities. The three things we import in large quantities are oranges, cherries, and onions. Yes. Now everybody says, "Oh, onions! I didn't know that." Everybody says, "Oh, no, onions." And the reason we've been importing onions for the last few years is because of the drought on the east coast, and we haven't been producing enough onions in Australia. So if you have a close look at your red onions and white onions, it's not the brown onions, you'll see that all come from the United States. We started importing them last month. We continue to import them until February, and then we don't import them. So onions, and we do import a little bit of pork, which is also a bit of a worry. But of onions, oranges, and cherries, what do you think might get contaminated with C. difficile? Onions, unless you've got a pig that's flying, and which of course is a possibility. Pooing on the onions and cherries. So my feeling is that this is contaminated food that's coming from the United States. And I've written about this, and I wrote this editorial in the MJA a couple of years ago saying, well, we shouldn't be putting cephalosporins into production animals. It's all going to come to a very nasty end. But this, of course, came out right. Yes. Does anyone know what was going on in the middle of 2009 when this came out? Swine flu. And, of course, we can't cope with more than one infectious disease problem at a time in Australia. So nobody read that. You write an editorial for the MJA, 24 journalists will ring you up and want to talk to you. But this was right in the middle of swine flu. Kathy O'Leary rang me up. I think she didn't want me to feel bad and said, can I talk to you about this? But that was it. One journalist rang me about this. So that's the problem. It's animals. So all these animals, that's a horse there with diarrhea. But so this is the most common cause of diarrhea in horses now. And it's all because of one drug. It's a drug called ceftiofur, which is the veterinary equivalent of ceftriaxone. And the vets love it because for the same reasons nurses love it. It's once a day. In fact, they now have a long-acting veterinary version of ceftiofur, which is called Exceed, and a 10-day course is 2 injections into a horse. So can you imagine how long? 1 injection lasts 5 days. So they love it. You may not know that certainly in the Northern Hemisphere, in the United States and in Europe, every egg that is going to become a broiler chicken, which is one that you're going to eat, every egg gets injected with ceftiofur. Doesn't happen in Australia, so we're lucky there, but every egg in the Northern Hemisphere gets injected with ceftiofur. And that's because they think, and you know, sometimes they're right, all their eggs are full of salmonella and they're trying to stop the salmonella in these eggs. But all they're doing is promoting C. difficile. So we don't have to worry about the chickens or the turkeys, but we do have to worry about all these other animals, I think, at the moment. So this is a paper from my good friend Glenn Songer from a couple of years ago, looking at meat in the US. So this is ground beef, ground pork, ground turkey. 40 to 50% contains Clostridium difficile for that reason. And it's these nasty strains. That same O27 strain I talked about, this one, which is in all the production animals in the Northern Hemisphere. So if you have a look at animals here in Australia, production animals, pigs, for example. As I said, Northern Hemisphere, since 2000, C. difficile is the most common cause of neonatal diarrhea, big economic losses. The interesting thing is that this strain in Europe, 10 years ago in Europe, this strain never caused human infections. But now it's the third most common human infecting strain in Europe. The problem in Europe is that people live right next door to pig farms. As I'm sure if you've been to Europe, if you go to the Netherlands, you can smell them. And you can smell the silage. So it's very difficult to try and tease out how people are getting infected. Whereas in Australia, of course, we don't actually live right next door to pig farms or cattle farms. They're way out in the country. So I think we've got a good chance here of actually working out what's going on. So big problem in pigs. And as I said, this strain now, third most common strain in Europe. And if you have a look at pigs, so this is piglets in Australia. It doesn't really matter which state you pick. The overall prevalence in piglets in Australia is 70%. So all our piglets have got C. difficile as well for the same reason. The vets are putting cephalosporins into these animals. What about cattle? Well, as I said earlier, if you're adult, no C. difficile in adults, or very low. These are also adults, probably adults that have had antibiotic. But if you look at very young cattle, so these are as I said at lunchtime to somebody, these poor little boy calves who, just because they've got testicles, they have to get killed because they're of no use to the dairy industry. Whereas in most parts of the world these days, bobby calves are not killed at age 5 days. They're, because that's socially unacceptable. The stuff you buy in the shops here as veal is not veal, it's baby beef. It's cows that are either 4 to 6 months of age. But we still kill in Australia roughly 1 million bobby calves a year, mainly in Victoria because that's the biggest dairy industry. And most of that meat goes into salami or small goods. So nearly 60% of these veal calves also colonised with C. difficile in quite high numbers. And if you have a look at the strains, and this is a bit small to read, but this is a ribotype 127. Now remember I just said that this ribotype here, which is 078, the one that's most common in the Northern Hemisphere, this one here is very close cousin, and all our cattle have got this particular one. And so these strains that we see in these cattle, this ribotype 127, and this one here, 033, somewhere there's another one that should be 126, oh here it is, 126. We're starting to see these ribotypes causing infections in humans now throughout Australia. So that's kind of why I'm worried. Now if you're a vegetarian, I know 1 or 2 of you might be, you're not really safe because all that pig effluent, big problem with the pig industry is it produces all this crap basically. And so they all have these big sewage treatment plants and they treat their effluent and then it goes to go on your rose gardens or your veggie gardens and they produce all this clean, what they call clean water, which certainly in Australia doesn't go on veggie patches because as I said, most of the pig farms are a long way away, but in Europe, all that water goes onto veggie patches. It's all full of C. difficile. We've looked at it, it's all full of it. So it doesn't matter if you're a vegetarian, if you go and, particularly if you go and eat organically grown foods, I suspect, and we haven't proven it yet, but I suspect a lot of organically grown foods have also got some C. difficile contamination these days. I was very worried about mushrooms, of course, because you're not allowed to wash mushrooms. You're supposed to dust them or something. I don't know if that dusting gets rid of C. difficile spores. Most, on the East Coast, all mushrooms are grown on, anybody know? They're actually all grown on pig litter. So they get all that straw and stuff that all the little pigs are growing. You're feeling unwell down there. So they get all this pig stuff, the pigs that are born on straw, which is going to be the norm because the little metal crates that everybody's upset about, they're outlawed as of 2017. So all piglets from 2017 onwards will be born on straw. Now there'll be plenty of deaths because the reason they're in those crates is so the mothers don't roll over on them, but on straw they roll over on them, and so lots of little piglets will die, be suffocated by their mothers. But all that straw is all collected up and then they compost it, supposedly, and then they wave it. I've seen it. They wave this sort of bit of steam over it, and they say, "Oh, it's sterile now," and that's what they use to grow mushrooms. We don't do pig shit in Western Australia. We use chicken shit to grow our mushrooms here, and as we know, none of our chickens have got C. difficile, so we're fine there. But if you're in the eastern states, I'd avoid the mushrooms as well. So our first theory in terms of what was going on was that, in fact, it was contaminated Australian meat and vegetables. And I still think that is probably going on. Driven by the flu season, we couldn't find this strain in any of our animals, and it didn't account for all the increase. And then, in fact, it was one of my postdocs, she said, this 244 strain comes from the United States. And that's when we got onto the onion theory. So we then discovered that there's another strain that suddenly appeared in Australia. This particular strain, very similar, same sort of scenario, community-acquired fear disease. Even by some tests, it actually groups with this O27 strain. It's so closely related. So what we're kind of finding by looking at these strains more carefully is that there are more of these nasty strains around than what we thought, because we haven't been doing this typing at all. So our second theory was that it's contaminated food from North America. We only import 4% of our food in Australia, so we're not badly off compared to many parts of the world. You may be interested to know that New Zealand has exactly the same food importation patterns as Australia, and they've got exactly the same problem in New Zealand with exactly the same two strains, ribotype 244 and ribotype 251. So I think still, though, that one of the contributing factors is the winter flu season, the fact that we have this seasonality in antibiotic use in the community, and now is about the time when you start seeing lots of post-flu secondary bacterial infections, and even not necessarily secondary bacterial infections, people getting antibiotics because they've got a virus. And there's the seasonality of onion imports into Australia. And as you can see, it's been going up. So since the drought, that was another drought period there, but since the drought, it's been going up. And so this 2011, 2012 was our highest ever. And then last season, it was about 50% of the onions. And very interestingly, the number of ribotype 244s we got in Australia dropped by about 50%. So the correlation is very good. I have to say I have not grown ribotype 244 or any C. difficile from onions, but we've only looked at maybe 3 or 4 dozen. We haven't actually looked at a lot because I kind of got onto this sort of earlier this year, the onion theory, and we stopped importing onions from the United States. So I've been waiting for the new onion season, which has just started. This is New Zealand. That's their onion imports. And I was talking to Sally Roberts about this at the beginning of the year. Sally Roberts being in charge of microbiology at the Auckland Hospital, saying, "Sally, I think it's the onions." And Sally said to me, "Oh, no, it can't be onions. We don't import onions into New Zealand. We export onions." I said, "Oh, I still think it's the onions." Anyway, next day I got an email from Sally saying, "I was preparing a spinach and risotto something or other last night, and I needed an onion. I went to my pantry, and there it was, product of the United States." So same sort of problem in New Zealand. Big, that same year we had big imports. They had big imports as well. And there's the seasonality of antibiotic use in Australia. This is the Netherlands as a comparator, much less, roughly the same population, much less in terms of antibiotic use. We use lots of antibiotics, and we have that peak, which is around about August, September every year. So roughly the right time to fit in with the onion imports as well. And so this is the, people laugh at this slide, but this is the correlation of onion imports and this ribotype 244 just in Western Australia. And so the dotted line is the onions, and we have no 244 till you get onions, and then goes up. And of course, we do get a few cases after we stop importing onions, but everybody stores onions. I know you put your onions in your bloody pantry for a couple of months, I suspect. I've got one sitting on my desk currently. It's been there for about a year and it looks just the same. And then even though that looks small, that's a 4-month period there where there's no 244 cases and no onions. And then as soon as we start importing onions again, the 244 cases come back. So I'd be very surprised if I'm wrong, but I have to say I haven't proven this yet. I think that's the problem. The other issue, of course, is the animal stuff. These are these nasty strains that we are starting to see in humans. I'm particularly concerned about this because none of the rapid diagnostic tests we have detect this strain. Because this strain doesn't produce that toxin A, doesn't produce that toxin B, it just produces binary toxin. And all our lab tests these days, except culture, all our lab tests, the rapid tests, are directed at detecting toxin A or toxin B, or the genes. So most labs won't be finding this strain. I'm particularly worried about haematology oncology patients because, and you know at Charlie's we have this big cancer centre these days, but hem-onc patients, you know, a lot of them are treated at home these days. They all, you know, all have lines in, but they're still in and out of hospital all the time. So there's a lot of C. diff in that group, but every time you have a case, everybody says, well, they get lots of antibiotics. That's, you know, they're going to be more susceptible. And they say, and it's always called healthcare related. And everybody says, oh yes, but they're in and out of hospital all the time. It has to be healthcare related. But when you actually look at the cases, you find that many of them presented at an emergency department with diarrhea. And then you look at the strain and you find there's a disproportionately high number of these strains that I know have come out of animals. They haven't come out of the hospital environment. They haven't. The hospital strains we have here in Australia are ribotype 014 and ribotype 002. After that, it just drops away. So if you see one of these strains, they've got it in the community. So if you look at what's going on in a little bit more detail, you can actually kind of make a little bit more sense out of it, which is what I've been trying to do. So it's kind of, this is a slide that a nice man named Pierre Formente gave me from WHO. And this is kind of the story of infectious diseases over the last 50 years. Most of them, 70% of the new infectious diseases over the last 50 years have been zoonoses. They're bugs that have come out of animals and are now infecting humans because we've either changed what we do with the animals or we've cut down trees in the wrong place and done all those sort of things that we shouldn't do. So they're, you know, animal strains that get into production animals, and then you get amplification, and then you get spillover into humans. And so if you go back to what I was talking about before with the 027 in North America, Ceftiofur was licensed for use as a veterinary antibiotic in North America in 1990. And in 2002, 3, they start having this huge issue in North America in relation to C. difficile. Ceftiofur was licensed for use in Australia in the year 2000. So here we are, 12 years later, we have this huge problem emerging with C. difficile. So I think what happened in America in the late 90s is what we're seeing now, and that is strains came out of animals, particularly that 027 strain, infected people in the community in North America at a time when there was no investigation of community C. diff for, of community diarrhea for C. difficile. So those people then front up to their small community hospitals. Again, they don't look for C. difficile. Then eventually, those people end up going into the tertiary system, which is what's happening here now. And then the tertiary system gets contaminated. And once the hospital system is contaminated, you've got this much more susceptible population of patients. And that's when you're going to start seeing people dying. So I think we're just repeating history, but of course there are many examples of history repeating itself. And I think that if we don't try and do something about this now, we're going to be in serious trouble. So I think this is a major new human health problem. We still really don't know that. I haven't proven it, but we've got to stop these nasty strains getting into the hospitals. And this is also an animal health problem, which needs to be dealt with. Lots of contamination, I think, outside the hospital, zoonoses, and it's going to require this One Health approach. And I don't know if you're familiar with One Health, but it's the concept that to deal with all these new problems, we need to engage vets and environmental scientists and a whole bunch of people and human medicine people and look at the problem together, because that's the only way we're going to solve this particular issue, and indeed a number of other issues that we've really created ourselves. On that note, I think I'm almost on time. I'll stop. Thank you. In fact, I've probably talked too long. People now want to rush off and have a large hamburger full of onions.
And salami.
And salami.
Yeah. Are there any questions for Tom?
I'm glad you asked because, well, two things. One is, it's those Spanish onions. Are there any Americans here? Well, I'm going to say something insulting. Spanish onions, which Americans think come from Spain, well, you know what they're like, a bit thick. So the Spanish onions, which are the ones you eat raw, are a real problem. The other issue is that what's called conventional cooking temperatures do not kill C. difficile spores. Now, if you get a big piece of steak and whack it on a hot plate, like you would for a barbecue, and there's spores on the outside, sure, that'll kill it. But, you know, a bolognese sauce is, technically speaking, if you do it properly, 50% pork and 50% veal. And you find that veal you buy for those sort of things and the stuff that goes into salami is going to be that bobby calf veal. It's not going to be good veal, and it's all going to be contaminated. So conventional cooking, whatever that means, that temperature does not kill C. difficile spores.
I once heard an American say the only way you get rid of it from the environment is with a blowtorch.
No, I think no. I think we are going to have to go, because as I said, to me, the issue is quite simple. You keep it out of the hospital environment, and then you can use whatever antibiotics you like. I think the way to do that these days is not to splash hypochlorite all around the place and make it smell like a swimming pool. I am a big fan of peroxide vapour, because I think that's had a huge impact in the UK in reducing their environmental contamination. Maybe, you know, depending on, if you've got single rooms, of course, it's very simple because you can just do each room. And I guess we're quite lucky at Charlie's. We've got lots of single rooms. And I don't know how many, the new Fiona Stanley, I think, as distinct from the old Fiona Stanley. The new Fiona Stanley, I think, has also got lots of single rooms, so that's quite good. So I think peroxide is the way to go. So, and I think C. diff's pretty well under control in the hospital system anyway. I don't think that's the problem. The problem's in the community, and the more we ignore it, the more we're likely to create a problem in the hospital system.
So how are you going to make people stand up and listen to those theories and just show up with answers and stuff? How long before we're going to start saying, oh, this seems to be something here and look at it?
It's a very good question. I posed that question to the Chief Medical Officer of Australia not 3 hours ago. I was on a teleconference. I think there's I think there is a change in opinion. I'd like to think it's because I've been knocking myself out, trying to get the word out there. But I think people are genuinely concerned. I mean, I go and talk to guys who run intensive care units in Western Sydney, like way Western Sydney, and during the peak of the 2 years ago C. diff season, as I call it now, they were saying that they would have a patient in intensive care, 2 or 3 patients in intensive care every week with severe C. diff disease. They weren't even sending off tests because they said it was so characteristic clinically, they were treating them straight away. I think that sort of word is getting out there and getting back to the people that need to make the decisions. And I can understand why, this is Chris Bagley, who's the Chief Medical Officer, has to go, there's a sort of a system that he has to go through. It has to go to this committee and that committee. But I think there is a change. I kind of feel there's a bit of a change. I certainly felt it today, where I think they're saying, well, we'll need to talk to you about doing a few projects to really prove the point. Because that's the problem. We haven't isolated it from food, but we haven't done much food culturing because, I mean, I've now got 6 postdocs and 6 PhD students working just on Clostridium difficile issues. There are so many of them, but we still, and one of them is doing food, but she's only just started. So. No, well, it's yeah, in the pigs, yes. Well, it's not well known. We've only just finished that study. It's certainly well known overseas, but it's not been shown until we've just done this study. And I'm going to Canberra in 2 weeks' time to talk to the pork industry about their problem. Now they're concerned, and look, the pork industry and the beef industry has actually been very proactive, much more so than the human industry, in trying to do something about this, because maybe it's to do with dollars. Pork's relatively small in Australia, but 40% of our beef goes to Japan and it's worth $2.5 billion a year. Now, if the Japanese people, who are really, anybody from Japan here? They're absolutely finicky about cleanliness. I don't know how many of you have been to Japan, but you can eat your breakfast out of the taxi because it's just so clean. Now, there's a few problems with that because you look at the normal population in Japan for C. difficile, and about 12% of them carry C. difficile. That's because they all take antibiotics prophylactically because they think that's going to keep them clean and healthy. So they have a much higher carriage rate of C. difficile than the normal population. But if they get an inkling that our beef has got some nasty bug in it, they'll stop importing it tomorrow. So I think those sorts of imperatives will cause the beef industry to do something about it, because it all comes down to money or dead bodies. And we haven't quite got enough dead bodies yet. But unfortunately, well, I'm not working on it. I'm trying to stop it. But that again is one of the problems. If you do stop it, everybody says, "Oh, no, it's not a problem." So I think if we stop putting antibiotics into animals tomorrow, it's still going to take 10 years to solve the problem, because the environment, the animal environment, is just so contaminated. So we go to all these piggeries, and their so-called clean water that they produce is what they use to clean out the farrowing sheds once they move. It's all in, all out, and it's all full of C. difficile. We've proven it. So it's not going to go away overnight. It's going to take 5 to 10 years. And in the interim, I think there's other things we're going to probably need to do. I think we're going to end up having to vaccinate production animals. So one of the projects I'm working on at the moment is producing a vaccine so that we can do that. It's not an easy fix once it gets out of control. But if you can get, well, that's the problem. So, the health departments, unfortunately, are reactionary, not proactive, even though that's what they say public health is all about, preventative medicine. I've talked to the public health people and they say, oh, they don't, I mean, the public health people, most of them haven't even heard of Clostridium difficile because it's a hospital problem, and certainly the concept of C. difficile being in food is very foreign. Now, if I went onto my slides or the couple of papers recently and I crossed out C. difficile and I put Salmonella typhimurium, there'd be hundreds of people running all over Australia investigating. And I talked to AusFoodNet about this, and they said, oh, it's not a notifiable disease, we don't have to do anything. So I thought that's what they were supposed to do, investigate foodborne outbreaks. So it's a little difficult, and I think it's very unfortunate that people are going to have to die before anybody does anything about it. But that's the world we live in, unfortunately. Yeah. That's I said, I think the infection control aspects of C. difficile are pretty good, or pretty simple. Is traditional hand washing. Yeah, soap and water. I mean, that's, you know, if they've got diarrhea, you do the same sort of things you would normally do with people with diarrhea. But for protecting yourself, just make sure you've got lots of soap and water. And I don't know, I'm always a little bit concerned. I'm concerned in my own lab when one of my workers or students is on antibiotics, because we've gone throughout our lab and you know, we're meticulously careful about how we deal with this bug, and yet we can go in our lab and do some swabbing of benches and we'll find it. We do find it. It gets everywhere. So, I mean, when you say, but have they got diarrhea? Are we talking about people with diarrhea or people without diarrhea? From a patient. She probably didn't wash her hands. No. Look, you know, the dreaded contact precautions. If they've got diarrhea, and wash. I mean, if somebody's got known C. diff diarrhea in their home, I would be giving them a bit of hypochlorite. I mean, I don't know what you would do, Marilyn, but well, no, I don't like hypochlorite either, but it does kill C. difficile spores, and of course, it, you know, it corrodes things and. Oh, sure, but we're talking about individual houses now. A bit of Milton, I mean, that's, you know, that's all Milton is. Sure. I mean, it's got to be clean to start with, absolutely. I think for the hospital system, peroxide vapor is the way to go to get your levels down.
I think the problem is hand gel physically.
Absolutely, and that is a real problem. And the other problem, of course, is now that we've spent years getting everybody to use alcoholic hand gels, it's sort of giving people mixed messages. When you say, oh, well, if it's a C. diff patient, you've got to wash your hands properly. So that's a bit of a problem to get across as well. But I'll let my infection control or infection prevention, what do you call yourself now these days, Madeline? Some fancy fancy names, but that's your job, getting that message out there. But you know, I think this is a big issue, and will be. How the patient treated? Oh, well, you can treat it with antibiotics. I mean, it sounds a bit strange, I know. Metronidazole or vancomycin, usually metronidazole first, vancomycin if you're really sick. And of course, the big trendy thing these days is a poo transplant, which I thought everybody knew about poo transplants. Yeah, God, best cure rate of the lot. I mean, it's an 85–90% cure rate. Put back the best probiotic in the world. And my feeling is that anybody goes to hospital we should have a bank and we donate. And no, seriously. And we freeze that, and then if you get your C. diff, you get a poo transplant. The FDA's just tried to ban it in the US because they said this is a drug. And they tried to make, no, seriously, they tried to make everybody that wanted to do a poo transplant, they tried to make them fill in an IND, investigation new drug form application, because they said this is a drug. And of course, you know, mountains of paperwork to do that, so everybody stopped doing poo transplants because the FDA said this is dangerous. and so then, you know, the patients just started doing them themselves. Because I mean, if you're, you know, 70 or 80 years old and you've had 12 recurrences of your C. diff, pretty debilitating, all you need is a blender and a piece of hose. You're laughing. And no, seriously, that's all you do need. And so then the FDA said, oh, this is bad because these people are doing it themselves. That's much more risky than actually having it done under supervision of medical staff. So they said, no, we can do poo transplants now for 2 blenders. That's right. So, but that's all you have to do, and it's fantastic. It works a treat. Patients respond within an hour. Within an hour, they feel better. So, yes.
I sort of was under the impression that you could have high enough colony, high enough count to be effective, and that you needed those ones.
No, I think the Valia is the one that's probably not quite high enough. So that's Valia yogurt, and you've got to eat about 30 tubs a day to get enough. But I thought Yakult, and I haven't actually looked at Yakult. We've done a bit of work with Valia. I mean, we did a study 15 years ago now where every patient, notwithstanding what I've just said about the numbers in Valia, every patient in Charlie's that was going on antibiotics, we replaced their dessert with a tub of Valia yogurt, and we reduced the rate of C. diff infection by 50%. And then the hospital said, oh, we can't afford this. I mean, it's ludicrous. No, seriously. They said, oh, we can't afford to do this. It's ridiculous because one case, you'd pay for your yogurt for the whole year. Just crazy. So that works. We treat all our recalcitrant C. diff patients with Saccharomyces boulardii, which is if you're a yeast taxonomist, it's exactly the same as Saccharomyces cerevisiae, which is, you know, brewer's yeast or baker's yeast, but boulardii is a different strain. And so all our recalcitrant C. diffs that have had multiple recurrences, we give Saccharomyces to. They get a week of vancomycin, 125 milligrams QID, with a gram, so 500 BD of this Saccharomyces for the first week with the vanc, and then they get 3 weeks of the Saccharomyces by itself. And we've only ever had one patient who didn't get better. We think she was a bit crazy, actually, non-compliant. So so that it does work. I mean, probiotics do work. And if you have a look, unfortunately, at some of the Cochrane reviews and systematic reviews, probiotics don't get a good rap for C. diff. Certainly for preventing primary infections, not good, but for recurrences, we swear by probiotics. Well, no. Yeah, Inner Health Plus, good. Yeah, we've looked at some of that. Everyone needs to go. We should stop.
Thank you, Tom. Thank you for your time, and thank you for very entertaining talk. Thank you.
All right. Anytime.